a deep dive presentation into molecular U. Um, the company is run by Rob. Rob Fraser is a CSO, president, and co-founder of Molecular U. As the as a founding CEO, he pioneered a low-cost method to analyze hundreds of biomarkers from a single blood sample and developed an LLM model integrating top scientific literature. He has expertise in molecular diagnostics, drug discovery, and regulatory compliance. He has led projects at Sanofi, Xenon, CDRD. Rob holds a PhD from the University of Alberta with postdoctoral training at Harvard and Sick Kids and IGBMC. He has co-founded multiple biotech companies and helped raise 37 million. Welcome, welcome, Rob. And we are excited to see what you have to share with us. I know a little bit about your company because you did share before, and it was very exciting. I thought it's a good idea to take a deep dive and look under the hood for especially for investors who are looking to invest in this company. Welcome.
Well, thank you, Lee, and thank you everybody for joining us this evening. I I really appreciate the introduction. Unfortunately, our CEO wasn't available today to take part in the presentation. He's traveling and preoccupied. But I've brought in our Chief Medical Officer, Murdoch Kalehi.
Oh, sorry, forgot to introduce him, but
Oh, that's
go ahead.
okay.
Please introduce Murdoch.
Yeah, because well, it's important we introduce Murdoch because he's going to lead the conversation today, and I'm actually going to hand over the reins to him. But I just wanted to thank everyone for attending. And Murdoch, did you want to introduce him, Uli, or do you want him? He can introduce himself.
I'm happy to. I'll
Go
just
ahead.
say, by background, I'm a physician and have helped launch a series of successful diagnostic technology companies over the last decade. And diving right into the presentation, I'll say, compared to any company I've ever worked with, I've never been more excited about when I learned about molecular medicine and its potential. And I've only learned about it over these last couple of years because it's only recent advances in technology, including with mass spectroscopy, that have enabled this practice of molecular medicine, which from what I've seen, is going to completely disrupt the way I practice medicine. But before I get into the details of the technology and the science, I actually want to tell you about Diane. Diane is a real patient of ours. Yeah, go ahead, Rob. The next slide. Diane is a real patient of ours that was getting regular molecular testing every year just to try and be healthier. So she was getting our standard panel of approximately 300 established known biomarkers to get a full survey of her health that looks at various biological processes and physiological systems and molecular functioning. We started to observe a trend around 2022 where a lot of those systems were starting to light up. They were not functioning as normally as they were before. And so we looked at all these systems and put it together and said, we are really concerned about cell proliferation and potentially some sort of cancerous process, and specifically looking at what's dysfunctioning. We're concerned about in the hepatobiliary system that there may be something. And she's like, but I feel fine. I got this test as my regular survey. And we encouraged her to follow up for further analysis. So she did get imaging of that area, which revealed a stage 1 pancreatic cancer. Now, for those who are familiar with pancreatic cancer, they know the usual course is for it to be discovered when you're symptomatic, which is typically stage 3 or stage 4. And that requires intensive surgery, heavy chemoradiation, and ultimately the survivability is limited. In Diane's case, because it was stage 1, it was a simpler treatment, and as you can see in her markers, she's been cancer-free ever since. A lot of people, when they hear that story, they get excited for Diane, and I am too. But what really excites me is we at Molecular U, we don't have a pancreatic cancer test. In fact, what we did, our process was simply looking at various systems and putting that together to assess for disease. And we wondered, could we do that with other diseases that are a product of such processes? So we took a look at some of the most notoriously difficult to diagnose cancers because they either are hard to identify or require really intensive and expensive screening to do so. And what we found is, so we took our models and applied it to these large data sets. It's retrospective. That's a qualification, but it needs to be when you're looking at large data sets like that. You can't do the prospective trials. And we found that by comparing sensitivity and specificity, the false positive and false negative rate at various diagnostic thresholds and looking the area under the curve, which is a surrogate for accuracy, with pretty high accuracy, we could identify these cancers. Now, is it perfect accuracy? No, but there's no perfect accuracy test. And if you compare these accuracy numbers to a lot of the standard screening tests, these do as well, if not significantly better, for lower cost or exposure, because this is just a simple blood test. And so even if they're not perfect, they certainly could be used to help raise or lower the probability of disease and be helpful in some sort of screening capacity. So this is cancer, and cancer was easy to look at because cancer is obviously this combination of dysfunctioning processes that can lead to this disease. But what about other diseases that are a product of progressive dysfunctioning that are difficult to diagnose and expensive to manage? So we looked at Alzheimer's. Alzheimer's is a product of various biological processes, whether it be inflammation or plaque formation or insulin resistance. And looking at that, we were, with a very high accuracy, able to identify and predict Alzheimer's. And I once wrote a book years ago on dietary interventions to help reduce the progression or reduce the risk and the progression of Alzheimer's disease. And the central theme of that book was Alzheimer's is, while it's a notoriously difficult disease to treat, there are much more effective interventions when you identify them early stage. But to do that, or to identify that risk early stage, but to do that, you need the tools to be able to identify the risk early stage. The molecular testing helps enable that. What I really love about molecular testing's potential for Alzheimer's specifically is Alzheimer's has various processes that can contribute. It can be a product of, like I said, inflammation or lipid dysregulation or insulin resistance. And how can you really prevent a disease's progression if you don't know the abnormal functioning that's causing it? Molecular testing, since it looks at those systems in what we call a pre-diagnostic way, a preclinical diagnostic, you can identify what systems are dysfunctioning and address those directly to help reduce the risk, rather than just try general interventions that you hope will help. So Alzheimer's is at one end of the spectrum, very late stage in life. Well, we wondered, what about diseases that come much earlier in life? That while they may not have the decades of biological processes that lead to them, they may have associations with various dysfunctioning biological processes. And again, like Alzheimer's, perhaps difficult to diagnose and often based on clinical symptoms. So autism was the perfect disease to look at. And again, very high accuracy numbers, because autism, while often diagnosed clinically, does have many of those biological processes associations. And we've gotten really excited about this because who doesn't want to better understand what's happening with their child and with their child's health? And molecular testing, this preclinical, looking at earlier stage, such as in childhood, allows for that. And earlier identification leads to earlier intervention, which leads to better outcomes. So autism isn't the only example of when we can take these diseases that are typically, diagnosed based on clinical symptoms and actually start to more objectively identify and quantify A lot of behavioral health, including anxiety, depression, PTSD, schizophrenia, while often diagnosed based on clinical symptoms, have strong associations in our biology, whether it be with specific biomarkers or entire biological pathways, such as neurotransmitter metabolism. Right now in clinical medicine, we don't have the tools to look at these entire neurotransmitter metabolism pathways and to try and use those to guide diagnosis and therapy. And instead we use symptoms, we give a drug, we have this trial and error. Let's give this a few months. We hope it helps you. We hope it doesn't make you worse, and we see what happens. I think that's not an ideal way to do things. With molecular testing, we have the ability to objectively identify, quantify the diagnosis of disease. And where I get really excited is then on the therapeutic side, we can start to identify therapeutics effectiveness. In fact, we did a small study. This is prospective, but with smaller numbers. Rob, on the next slide, where we took some disease processes, carcinoma and melanoma, that were getting interventions, getting treatments, and seeing if we could assess the effectiveness of those treatments. Because we recognize in medicine, a lot of it is trial and error. It's based on these large studies that over large populations have some effectiveness, but we don't necessarily know how it affects the individual. But by doing molecular testing, we can very quickly, much quicker than just waiting months to see if the treatment has effectiveness clinically, we can very quickly look at that preclinical level and see, is the drug having its intended effects, whether in behavioral health and correcting neurotransmitter metabolism, or in this case, reducing inflammation and improving lipid metabolism and reducing complement activation. Now, I'm excited about that because, again, earlier intervention means less time where you're taking a drug that's potentially ineffective and costly, but it also reduces the risk of side effects. In fact, we wondered, could we use molecular testing to help assess adverse events? So in that study, where we had people who were on these immune checkpoint inhibitors for cancer therapy. Immune checkpoint inhibitors are notorious for activating the immune system, and you can go on to develop rheumatoid arthritis or inflammatory bowel disease. And so we thought, hey, can we identify the risks of those adverse events earlier by looking at the molecular level? And so we performed an analysis, and it turns out we were able to identify adverse events 2/3 of the time. But here's what gets really exciting. That 2/3 of the time we were able to identify adverse events, that was before the therapy was even started. Imagine being able to know ahead of time, before even coming close to developing another debilitating condition like rheumatoid arthritis or inflammatory bowel disease, being able to say, hey, perhaps this isn't the right drug for you, and we can adjust to this other drug, which may be more effective and may have less adverse consequences. So at this point, I've talked about a lot of potential uses of molecular testing. It probably feels like I've suggested it can do everything, and it can do a lot. And we'll talk more about that. But I think what fascinates me about all these potential uses that I've talked about is, again, like in Diane's case, where we didn't have a pancreatic cancer test, we didn't have specific tests for any of this. We didn't have specific tests for any of the other cancers or autism or Alzheimer's. We're looking at this, we're looking instead at the biological pathways and processes. And instead of selecting for a different test for this person because of this risk and this person, we performed this exact same test on everybody. So all of these applications came from the exact same test. Now, as a physician that has spent many years learning how to discern symptoms and be very selective about the diagnostics I choose, this actually initially felt a little uncomfortable for me. Like, you mean I can't take all those years of knowledge and and be very discerning, and instead I'm just doing the same test every time on everybody. Um, and what I've ultimately come to is accepting, realizing that while as a physician I am very used to practicing in a discrete and reductive way, focusing on specific disease processes, that's not actually what patients care about. What patients care about is I want to be healthier. I want to live longer. I want you to look at all of me. And that's what the molecular testing of hundreds of biomarkers that cover all these comprehensive systems that are inextricably linked in many ways. Many biological processes are associated with many organs and many diseases. That's what we ultimately want. And we ultimately want this to happen before we develop disease and not actually later stage when it's much harder to treat. So with all these applications, like I said, this probably feels, like it seems too good to be true. One test that can do so many things. Again, I felt uncomfortable when I was first learning about all this. And whenever we feel uncomfortable, our mind comes up with counter arguments of oh, why this can't work. This can't actually be true. So I'm actually going to share with you some of them. Some of the ones that my mind came up with. And the first one was very obvious to me. Like, you're telling me you can do all these things and analyze hundreds of tests with a small volume of blood. I think I've heard this before. I remember when I was working with one of my first diagnostic technology companies, and Theranos was the big elephant in the room that was going to take us all down because of what they can do. And we were very afraid. So when they started finally offering some of their tests at some Walgreens clinics in Arizona, our company actually sent some people all the way to Arizona to those Walgreens to get the Theranos test just so we could understand what we were up against. And the most interesting thing would occur every time we sent someone in. The phlebotomist there would say, oh, actually, we can't do the traditional Theranos draw on your tests. We have to do a more regular, blood draw. And this happened every single time we sent someone to get the Theranos test. So we knew something was up. I remember I was on a conference call shortly after this, and I said, I wish this company were publicly traded so I could short them, bet on their downfall. And then months later, we all know the story. Unlike Theranos, Molecular U is not just saying we can start doing this. We have performed this testing on thousands of people over years. Diane is a real case. A lot of the other cases Rob are going to tell you about are real people, real research we've been doing. Also, unlike Theranos, that shrouds itself in secrecy, we go out of our way to talk about our testing and our science. And so Rob is going to tell you more about our testing and our science, and we'll eventually come back to some of the other counter arguments that the mind naturally does. Thanks, Rob.
Thank you, Murdoch. So going back to some of what Murdoch said is what we began really as this as a big data exercise, and the big data sources we use with the world's literature to land on those high value metabolites and proteins that can do exactly what Murdoch was mentioning and detect disease at its earliest stage, get into the mechanisms that drive it, and focus on those functional elements of our physiology, our biology, our pathophysiology, so that we can get in front of disease early and have the best possible outcomes. And so we ended up developing a first version of a metabolome proteome combined assay. We run everything on well-known technology, LCMS. They've been around for years. We didn't invent these instruments. We just take advantage of the advances that have been coming along. They're sampling faster. They're able to give us absolute quantitation. We can sample because we're using mass spectrometry. We can multiplex on the same platform, which gives us this great advantage of being able to compare all of these values using the exact same technology. So I'm not jumping from an immunoreactive test to a light-based assay to a radioactive assay. It's all done through one platform. And then the other advantage to the mass spectrometer is that it gives us absolute values. And those absolute values are a great starting point for any calculation. And so we've used our AI tools that identified them to also curate all the world's literature so that we can make sense of what those values mean and run it through our algorithms in our clinical support platform so that we can then rank order the risks for an individual based on the associations with the diseases, similar to the pancreatic cancer. We can break them down into pathways so that you can truly understand what the root cause of the disease is, so that becomes the focus and the energy to help the patient get better. And then we support along the way with lifestyle action plans more on the diets that would be better for the individual that can affect or known to affect the biomarkers, and exercises and supplements. We've worked hard the last little while. We've just launching now in the US. We've been around in Canada in the market since about 2017, but we've now focusing in the US, and we've had to set up a lot of our logistics. The first one really was to focus on a lab that would work with us and run the assay in a CLIA environment, so we could run it as an LDT. So that's now most of the way there. We should be done next week. We had to get the sampling. So one of the challenges we have working with metabolites is that they tend to be a little labile. And so if you don't freeze them within a couple of hours, you'll lose a lot of the integrity of the sample. And so if we want to get that full insight and the full understanding of what's going on in the individual, we have to have this cold chain custody in place. And so we had to once get the phlebotomy worked out, make sure that gets shipped to our labs on ice cold. It has to be down at -20 and stayed at least frozen until we get it to the lab, and then we can analyze it. The lab then runs the assay and pushes the data into our platform. We process it we're using our algorithms, and those algorithms deliver it in a very easy to use interface that the practitioners then work with. And this is most of what it looks like. We've sort of scrunched it down into one slide so you can get a sense of what's going on. So it starts with the health areas being ranked, ordered, a low score being a high risk, a high score being a low risk. You can click on any one of these and you can go deeper into each of the biomarkers associated with the condition. The pathways are broken down, also ranked in order of how perturbed they are, how many values are out of range, and which ones are most important, particularly as they apply to the health area affected. And then you can break down each one of those into the individual biomarkers. These are all dynamic biomarkers, so they can be modified, and they can be modified with medicine, with lifestyle changes. All of these things can affect their levels, and they can be stabilized. And so the key is to follow these biomarkers over time. And so get like with Diane, when she's healthy, we had a very clear picture of her health. Then when the pancreatic cancer occurred, we were able to see it at its earliest stage. And so that longitudinal monitoring is very important, and monitoring these biomarkers is key to people's health. But it's not just about the biomarkers, and as Murdoch mentioned earlier, it's about figuring out the pathways. And because if we understand the pathways, that allows us to deliver a very specific care path. And so in a small pilot that we initiated with a clinical lab partner out of Florida, we analyzed 80 patients. 5 of them were diagnosed with Alzheimer's disease. About half of them had early signs of mild cognitive impairment, and then the rest were mostly healthy. And from there, we were able to analyze which ones were at risk for Alzheimer's disease. And it was fascinating that we were able to see we were able to stratify the population into 3 endotypes. Pardon me. And these endotypes then allow us to focus in on which care path is going to be most effective, and we can follow that more specifically. So it really gets into that personalization of the care. So we're a company, and we're focused on coming to market. Our goal is to first work with the concierge and integrative clinics. They're very familiar with a lot of these longevity type tools, and that's been a big focus for us, and I'll touch base on that. We've talked a lot about the neurology that where we can support neurology, and we have a large partner in Buffalo, the Dent Neurologic Institute. It's actually the largest neurologic institute in the U.S. And we've got very excited about the behavior health, and Murdoch shared some of that with you. I'd like to talk a little bit more about some of the other areas, but we also have a reimbursement model that we're working on, and so we do feel that this will be reimbursable by 2026. A number of the metabolites and proteins that we quantitate have CPT codes, and so with a stacked billing or a super bill, we feel that this could be reimbursed probably for about half of what we're charging. So we do well in longevity. We had a proud moment in at Health this year. We got first prize, shared first prize in the Digital Health Hub Foundation Awards as a rising star in the longevity category, primarily for this concept that by getting in front of disease, identifying disease early, you have a much better chance of keeping disease at bay and thereby living a longer, healthier life. We also feel there's a great opportunity in supporting practitioners and patients in weight management. There's a lot of excitement and understandably so around the GLP-1 agonists. But there's been a lot of claims and most of these are done on a population based and that's how things are reported. We don't get that individual data. So how will you know if your patient is actually responding in a positive or negative way if you don't have the information? So if you can take a baseline prior to treatment, then follow up 2 to 3 weeks later, you can see if you're affecting those biomarkers that are indicating, hey, this is doing great things for your central nervous system. Those risks for Alzheimer's disease are actually attenuated. However, we're getting muscle wasting, so we need to figure that out. We don't want you to lose muscle while you're losing your weight, your fat weight. And if we're seeing things going on in the pancreas, like pancreatitis, which has been shown as an adverse event, we should be able to pick that up even perhaps before the treatment begins, because they might already be predisposed to that particular adverse event, similar to what we saw with those receiving the checkpoint inhibitors. We also feel that this is long overdue, a system that can truly monitor women throughout their lives. I mean, women have many more changes that they go through, and I'm not speaking from experience, but from a scientific perspective. But I think these are really important to understand because we're really blind. There's no model in the animal kingdom that we can use to study how women go from normal menstrual to menopause to postmenopausal, and what are the health risks associated with it. We actually have the tools, the platform that can give that insight and manage it. And even when interventions like an estradiol replacement are suggested or given, we can monitor to see if that increase or lowers risk across all of the areas you see here. And even with younger women, we can support them in with the management of polycystic ovarian or the early detection, so they don't actually suffer any infertility. We got particularly concerned about what was going on in LA, and it raised an alert for us that many people could be exposed to these wildfire effluents, and what are the things that we could do as an organization to support these people or those on the front lines, our first responders, which are becoming more and more active members of our society and thereby that much more important to maintain their health. Wildfire can affect so many of our systems from the effluents that are coming out, many of them directly toxic. And some of them are very well documented, and we know that we can monitor the neurological and cognitive effects, lung and respiratory damage, the metabolic dysfunction and oxidative stress. These can also lead to coagulopathies, leading to cardiovascular disease. And, as we eliminate the toxins from our system, the kidney and the liver can have damage as well. And then overall, there tends to be a very strong inflammatory response as these various toxins enter our system. So we launched a project with some, the other area that we did touch on was the cancer. And so one of the things that is known is that first responders tend to be at a higher risk, particularly firefighters at a higher risk for cancer. And so we all recognize that cancer has a huge genetic component, but it's not really the most common cause of cancer. It's usually the environmental exposures, and that aligns well with what the firefighters might be exposed to. And even things like pancreatic cancer, there's very few inherited patterns. And so what tends to be the greatest cause of cancer is the environmental exposures and aging, and aging is really just the more time we spend being exposed to these potentially carcinogenic factors. What we can measure are all of those key hallmarks or tenets of cancer that were listed, some of them with Diane, but we also are able to look at mutagenesis to see if there's factors in our blood that it can actually cause the mutagenesis, in addition to all of the others ones listed here, which I won't go through. So we did, this is the second case study we'll give you today around the cancers, and so we have started a project with some firefighters. And protecting this isn't the individual's true identity, obviously, and he's somewhere in the age of 40 plus. And he's at that age, he's very disproportionately affected, and we actually noticed the whole population had a disproportionately high level of risk for type 2 diabetes and cardiovascular disease, as well as rheumatoid arthritis. And this individual actually was very much aligned with the population. But what struck us about this individual is that a number of the other biomarkers that were pointing to these key cancer associations are also out of line. Now, we don't know that he's in the same place as Diane, so we are going to be monitoring closely and doing another test and supporting him, because these are still very early stages. But it does appear that there is some neoplasia beginning here, and that this is another example of how monitoring people at risk in particular can be a very effective way of getting in front of cancers prior of them becoming advanced and in the advanced stages and harder to treat and much more expensive. The other thing that we've been doing is, so we currently have a chipset, as we're calling it, for about 280 metabolites and proteins. We're always upgrading this, and we're going through the process of curating and identifying new biomarkers to put into training our algorithms and validating things in the wet lab, so that we can get them into our next panel and multiplex them. Our goal is to get to about 800 metabolites and proteins by the end of this year. But in concert with that, similar to any sort of chip, like an NVIDIA chip, if you like, we need that software to support it. So at the same time that we do the curation, we have to get those algorithms we're using a lot more AI tools now too, that makes our processes and deep dives much more straightforward, to provide those insights and analytics that'll be pushed out to the practitioners, and then continue to expand and on the data side of things, so that we can continue to learn and apply those learnings to the practice. And then the last advances that we've been working on is really employing large language models to summarize, because it is a lot of information for anybody to digest, and our scientists use these tools all the time. So why not make them available to the practitioner? Currently, they're heavily supervised. That's why we call them AI assisted. We still get a lot of hallucinating, I think is the word, when you the large language models are actually fabricating information in a very clever way. In fact, we've been fooled a couple of times and had to cross-check and make sure that the references are actually real. But the goal is to make it nice and summarized, and eventually we'll be able to put it out so that practitioners can query it and go even deeper with their own inquiries. So I'm going to pass it back to Murdoch to go on with his counters.
Well, thanks, Rob. So, the second thing I think when I think of something that does hundreds of markers is that it must be too expensive. In general, when I look at the healthcare system, what seems like, if we feel the healthcare system is too expensive, it seems like the only way we're going to reverse that trend is to come up with higher value interventions, things that can do more for a lower cost. And I believe molecular U is one of the highest value interventions I've seen, because, I went through all the different, potential applications it might have, or actually I went through a subset of the potential applications, all with the same test, a single test. And the cost of that test to run, it's in the hundreds of dollars. So one test that for a pretty nominal cost compared to most healthcare interventions that can assess for so much. And that cost is pretty comparable to most screening tests if you've ever gotten a lab bill, except unlike those tests, which seem to be only getting more and more expensive, the molecular U testing, because of innovations in the technology, are getting cheaper. As Rob mentioned, by the end of the year, we expect to have an 800 marker panel. And we've talked about panels further down the line that have thousands of markers without a significantly increased cost. So the cost per marker is decreasing exponentially, like with a lot of technology. But I think the biggest cost savings is simply comparing it to the alternative of not identifying or addressing the disease early. If you think about Diane's case, what did she avoid? She avoided much more intensive surgery, much more intensive chemoradiation, potentially death, or at least much more severe morbidity, all for a test that costs hundreds of dollars. I think that's the higher value intervention we're looking for. The next counter argument that people think of when they say hundreds of markers is, I and many of the people on this call are physicians. Like, I'm already really busy. I can't interpret hundreds more markers. And that argument's absolutely correct. If I just gave you this list of 100 markers with the values, especially as these panels continue to grow, no one could decipher it. And so we have spent as much time on our analysis technology as on our technology to help make it understandable, to take these hundreds of markers and, as Rob showed some of the user interface of the platform, decipher it into where are your highest risk markers and biological processes, and what are the potential interventions and action plans you can have. And we're even recently started using LLMs to help translate this into a short narrative summary. With the most recent version of the product I've seen, you can understand everything you need to know in terms of where the risks are and what steps you can take within 60 seconds. And then you can dive deeper and further into areas as you would like, if you'd like to spend more time. The final counter argument I sometimes hear is, and because we encounter this in medicine sometimes, you do more tests, you're just going to find more things, and we're going to end up chasing more things. And it's going to be like a PSA. It's going to be, oh, okay, well, now we have to do this follow-up test, or follow up this mammogram, and it's actually just going to raise costs and potentially not have a huge impact. But that's actually the beautiful thing about this method of this preclinical testing hundreds of markers, because when you can test that many independent data points, you're no longer chasing individual markers. When you have hundreds, you're not going to chase one. You're looking at entire systems. And when entire systems are dysfunctioning, that tells you something's really going on. That's why we have those high area under the curve numbers with that high sensitivity and high specificity, or low false negatives and false positive. Because if something is showing up, that means it should be chased. In Diane's case, it wasn't a marker we were chasing for her pancreatic cancer. As Rob said, it was 56 of them. So when you have that many areas of dysfunction, you should go after it, because, in Diane's case, in the next slide, Rob, doing that is what actually saves lives, because by enabling early detection, you can do early intervention, and early intervention is what saves lives. In Diane's case, because molecular U saved her life and continues to monitor her life, she is a customer for life. So I think that's the last sort of telling thing about the value of molecular medicine. Anyone who's had it, anyone who hears about it wants to get it, but anyone who's gotten it, who's had it, wants to keep getting it. So if people on this call want to learn more about how you can potentially access it, Again, unlike some other companies, we love talking to other people about our technology and about accessing that technology. So, there's people on this call. You can follow up with us, and thank you so much for letting myself and Rob talk a little bit about how molecular medicine is going to completely disrupt and change the way I practice medicine. Thank you.
Thank you.
Thanks, Murdoch and Rob. Great presentation. I'm sure there are a lot of questions coming in people's minds, but please raise your hand if you want to ask a question, so that I can unmute you and get you to ask. So I have, I was looking at this, and then two things pop up for me. And of course, all those arguments, I agree that, prevention is probably better than cure. Um, one of the things I'm noticing is that you can actually, and we did that. We did one study with biomarkers for diabetes. One of the things that what you shared was that you can actually subdivide subclasses of a disease based on how the markers are showing up. And so it's not, type 2 diabetes is not one disease. It is probably several subclasses of disease where, if there are 70 plus drugs to treat diabetes, which drug should that person get? And that gets a little tricky, because if you don't find the right drug, that means that the person will have high sugar levels for a long period of time. And so I'm thinking that becomes very powerful, especially in this case, Alzheimer's and maybe autism, maybe, all these big blanket diagnostic groups. You can actually separate them, tease them out based on their pathophysiology and based on their biomarkers. Do you want to comment on that?
Yeah, you want me to take that one, Murdoch?
Yeah, please.
Yeah, so absolutely. I mean, I think you're right on. I think we're just scratching the surface with the Alzheimer's disease. I think we'll probably find some other endotypes that people can get Alzheimer's disease by. And if we treat them all the same, we might just be not hitting the mark, right? And so you really want to be focused in your care path. So this enables that focus. And so it's interesting. We only advise on lifestyle changes. We're very interested in going the next level at some point, and how we can support on the use of medications, but that's outside of our wheelhouse right now, just because of where we are in our regulatory path. But I think you've hit it right on, Newly, and diabetes is going to be the next one to start to break down to figure out where the interventions will occur. This particular cohort just popped up. It was quite elegant, and one of the things that's quite interesting is that 2/3 of the Alzheimer's disease population is female. One of these groups actually was all female, and so there might be a bias for women in a certain path, and we don't know what else is contributing to that bias yet, but that's what we'll be continuing to understand as we increase the numbers in these cohorts.
Yeah.
Charlie?
The second point, and Charlie's coming up next. The second point I wanted to make was that if you're treating a condition and you're tracking the biomarkers, is that always following the improvement of the condition? Or, I mean, can we safely say that, oh, the biomarker is improving, so the patient must be getting better? Is that true in every case, or is that not known?
I think it's been true in every case that we've examined thus far. And if you think about diseases being a physical thing, like our holler systems are made as being a physical thing, what's driving those, what's shifting those is what we track. And that's most of what our medicines have been focused on, right? Is what are the driving forces in disease that we need to intervene with to block that from progressing. And so it is all about getting in front of these pathways, understanding the reporters are, and most medicines you have are symptom based, and being in a pre-symptom base, we have to rely on those biomarkers because there are no symptoms necessarily. But
Yeah.
when we did the study with the cancer patients and we saw the attenuation of the biomarkers with the treatment that coincided with the improvement of the patients, we also with the rheumatoid arthritis and the inflammatory bowel disease that was exacerbated with the treatment. And so we could see those too, and it was all based on the biomarkers. We actually didn't know who was who in the analysis.
Yeah. Yeah. I mean, because I can see how this can be very useful in cases of mental health problems, because
Yeah.
the symptoms may not, for example, suicidality, how do you measure? There's no measure for how suicidal a person is, or how
Correct.
how that's going to change the outcomes. But it's great. Charlie,
Yeah.
go ahead, unmute yourself and ask your question.
Yeah, fantastic presentation, and equally impressive as my first discovery of that at the Personalized Medicine World Conference, or PMWC, in Santa Clara last month. And particularly of interest to me because my wife was diagnosed with pancreatic cancer stage 4. And so I'm now on a new journey to find ways to diagnose it earlier. And that was the reason I was looking for your company at PMWC. So I have a series of questions. Try to they're aligned with the applications that I felt needed to be done to transform how we care for cancer in general, with you know those cancers that are difficult to diagnose, that are symptom driven, and often late and delayed diagnosis. So first question is, this is a huge transformative change, as you've described, in contrast to our usual way of using diagnostic tests. We think of, a diagnostic test that has a certain sensitivity, specificity for a given disease. I went to a conference on early detection of cancer last year, where one of the scientists who had been working in the field for a number of years said, we're not making progress. We are spending 10 years and thousands or millions of dollars, and we're not getting tests that really help patients. So, what's the regulatory path, if any? Because I think that's been one of the big barriers.
So our so we're operating as an LDT currently, and I think this particular example of Diane, where we can support an individual in managing the early detection and avoidance of chronic diseases, which was the original use case, we can pick up these larger disruptions in biology much more readily because we're doing it on a regular basis. And so it's got this two-side, double-edged sword, if you like. So on one side you can look after the chronic disease, and on the other side we can take on these cancers that can appear, and none of us really know when or why they're going to appear. So monitoring the biology is really the key. So while we're treating this as a primarily longevity test, keeping people healthy, when we see a perturbation like this, we alert the practitioner, and the practitioner is really left to decide if they treat this as a vital sign and they want to move forward and take it a clinical path, as in the case of Diane, or do they want to say, well, let's do some more testing, or we'll I don't believe this, or what have you. So we were fortunate, I think, in Diane's case, that the doctor was very proactive and treated this as a vital sign, and then went the next level and had the imaging ordered, and then did the biopsies shortly after.
So
Yeah.
to leave more time for others, I don't want to monopolize the time, but we can follow up later. There's a series of applications where I think this could have big transformative effect, and they may need studies or research done to demonstrate or prove them.
Mhm.
And I'm very happy to discuss those with you. I had experience within pharmaceutical companies at Pfizer, Roche, and Genentech, and would be happy to follow up with you later.
That'd be great.
Edward is next. Edward, go ahead. Ask your question.
Well, I really appreciate the time for the question here. So I work in the chronic pain space. So as you know, chronic pain there's a lot of people in chronic pain. It's a big consumer of healthcare resources. And I look at this here as an application here is in terms of the chronification of pain, right? Because I think in our lifetimes, we all experience some degree of pain, all right? But we all don't develop chronic pain because of it. But there are certain cohorts that do develop chronic pain. I think this application looking at these different systems that, in my brief view, do have some involvement in the chronification of pain symptoms.
Mhm.
I'm also very curious about, well, I know that there's a relationship with mental health and the chronification of pain. But again, I guess my question is to you here, it'll be a very interesting comparison to look at those individuals who report chronic pain versus those individuals that are reporting pain for the first time.
Mhm.
Which systems are different, light up compared to those the first time? Very interesting. Give us potential targets, gives us potential interventions for preventative health, for lifestyle modifications, for focus on different types of mediators to prevent the chronification of pain. But that was more of a comment. And if you have that data, I'd love to see it. Or if you have that, that interest, a small work group or something to look at it there, because I'm sure you probably have it testing, having done this for many years. We'd love to take part of that too. Thank you for your time.
Thank you, Edward. And I'll just I've done a lot of pain research myself, and actually did work. You're probably familiar with congenital indifference to pain. And we identified the gene responsible for that. And it was fascinating because we got a very safe and effective and specific drug that was a sodium channel blocker that was very specific to this one pain channel that was associated with the congenital indifference to pain. It was the Mendelian inherited gene, so it was a complete knockout, loss of function. And when we got in the clinic, it actually had pretty modest effects, and it just highlighted the complexities of pain and how experiential it can be and how many confounders it can be. There's inflammatory pain. There's so many different differences, and so I agree. This is a great platform to start to decipher what sort of pains are involved, where might the inventions be more effective. Would love to explore that with you.
Right. Let's see. Ramana had a question or comment. Ramana?
Yeah. I think, I trust Canadian companies anytime. I lived in Canada for a while. But I want you to connect with Roger Royce, is my attorney, and he's actually publicly announced it. He was diagnosed with pancreatic cancer. It's one of the most difficult one to diagnose, right? So I saw your chart. I didn't understand a whole lot of it, but I think you're onto something. I think that is the one area that is where big help is needed. The pancreatic cancer. That on the y-axis, I don't know what biological processes you're mentioning. So yeah.
I mean, this is the biological processes are really the driver, the various processes involved in either the inception of cancer or the mutagenesis. You see a lot of inflammatory responses as our body actually tries to defend against it. And then you have angiogenesis, which is feeding the tumor. We were actually quite surprised when we found out the stage of this cancer, because when we looked at these processes that were at play, they seem very advanced. And so it just gives you a sense of just how aggressive cancers can be at those early stages. And this may be a hallmark of pancreatic cancer and why it's so hard to treat, because right from the outset, it is basically setting the stage for success as a cancer. That's just a hypothesis, but when we see these kinds of markers that are kind of checking all the boxes for the cancer to really be successful, at such an early stage, and it was a very early stage one. So yeah, we're excited, but I think the key really is this longitudinal monitoring, so you can get it as early as possible. If she started in testing in 2024, we probably would have found stage 4 pancreatic cancer.
Yeah.
So
Yeah.
I think it's really important that it's happening on a regular basis.
Yeah,
Thank
he's
you.
looking to support these companies like you, just connect. I send any email. Dr. Uli, I gave an email up there. If we can
Sure.
pass that to them, and yeah.
Actually, the survey is going on, so you can put that email in there.
Ramona?
So Susan is coming up next, but I was thinking maybe ovarian cancer may be a good candidate too, because that
Mhm.
gets diagnosed late and
Yeah.
because the symptoms are hidden. So
Yeah,
that's
we
something
have
that may.
ovarian is actually one of the ones we couldn't fit it into the slide, but we do look at ovarian. We've got kidney now, and we're looking at non-small cell lung carcinoma, which is
Yeah,
also very late. Like lung
that's
cancer is
hard.
notoriously late. But yeah, and maybe breast is being more easily monitored now, but to combine that with a blood test and maybe a mammogram, we'd have a much clearer idea of what's going on with the
Okay.
individual.
Maybe it could replace mammogram. You don't
Mm.
know. Susan, go ahead. Susan has multiple questions, so
Oh, no, I'll make it short, but I just want to know
no, take your time.
how you came up with this. What research did you study? Or what researchers did you speak with that came up with these biomarkers? How did this come about?
How did it all come about? That's a great question.
And how is this FDA approved? Are you planning to get that? And also, it doesn't seem that very expensive or concierge medicine at this point, right? Doctors are using it and they're giving you their data at this point when they use it. Are you doing studies?
The EHR data, you mean? They're
Are they giving you data from their practice to help
Yeah.
you?
Yeah. So okay, I'll start with a bit of the background, and then I'll try to answer the other questions in the order that you've asked them. When my background is actually in the pharmaceutical industry, most of my career was focused on just working out the mechanisms of disease, and I used a variety of technologies, everything from genetics to mass spectrometry, proteomics, metabolomics, and most because it was more focused on finding that those factors that we could intervene with, it tended to be somewhat limited. And so I teamed up with one of our more prestigious scientists in Canada, a fellow by the name of Dr. Peter Cullis, and I started this thing called the Personalized Medicine Initiative, which was really an effort to go beyond just treating the disease and treating the whole patient, understanding what the patient needed. And that led us to examining all of the various technologies that we could apply to an individual to get ahead of disease. And so we used whole genome sequencing, epigenetics to see which genes were switched on and off. We looked at microbiome, metabolome, proteome imaging, and we combined it all. And we really found that what was most insightful was the metabolome and the proteome. Because it was what was happening right now. The genetics was useful, but it was only primary sequence variants that you didn't know when in your lifetime these things might manifest or if they would manifest. The epigenetics wasn't really that actionable. The microbiome was still a little bit early, and we've learned to look at the metabolites that are generated from the microbiome that are actually absorbed into the system and do have profound effects on our CNS function as well as cardiovascular function and kidney, we're learning. And so it just became very obvious to us that it was cost effective, and the advantages are that you can multiplex these biomarkers on the platform. And we were fortunate enough to have two individuals in Canada that were world leaders in this space. Dr. David Wishart, who's led the human metabolome effort, which was similar to the human genome. So we had a great start with him. And then we also teamed up with Dr. Christoph Borhers, who's a leading proteome analytic chemist, and really enabled the MRM and multiplexing that we utilize for the proteome. So that's how we had the foundational science, and then we had to build all the rest, make sense of it all, and that was a big effort from more on a using at the time it was more natural language processing and digging deep into the literature and finding what metabolites were most important, what do they mean, how can we go further with them, how do they combine, and so it's been a beautiful exercise, and then simplify it so that people can actually use it. So it's been a huge effort and taken a lot of really great people to pull it together. The second question I've already forgotten was
Or
The FDA.
FDA path. Yeah, so the
FDA.
FDA path. So we do have a path where we don't need, so we're operating as an LDT. We don't know what's coming next. We know they want to eliminate LDT, so we might have to operate as an IVD. But our
Can you explain
advisors,
the acronyms?
oh, sorry.
Expand that.
Lab developed test is operating in a CLIA or a CAP environment that's the lab is regulated and the assay has to be run under certain conditions. Whereas an in vitro diagnostic is a FDA approved test that has to go through a series of trials, et cetera, to get the full approval. So we base everything that we've got on the world's literature. So there are a lot of work that's been done. It's all done in humans, and so we reflect that information on the individuals. So that's how we're able to get so much information down into one place. The if as long as we don't prescribe and we use language in our so we do health risks, not disease risks. We don't need to get FDA approval. We're clinical support. So as long as we're working with clinicians in the practice of medicine, we don't need the FDA approval. However, saying that we do have a pathway and we're going to do a key submission to the FDA to start the approval as a de novo device for a class 2 medical device, software as a device.
I
So that's
assume
our
you,
path.
I'm sorry. I assume you have a patent, and who are your competitors?
So we do have patents around the platform and some of the signatures that we shared with you. Uh, there are competitors mostly in most groups tend to focus either metabolome or proteome. There aren't too many doing what we're doing. Um, I don't think there's any direct competitor currently, but there are similar efforts. People have pointed to function, but function is a really interesting approach to taking a sort of a shopping cart and all the different tests you want to get and work with your doctor to see which ones in a direct-to-consumer model. They are actually a frenemy of ours. They're a group that we would love to offer our tests through. So we don't see any direct competitors right now. There are other multi-omic groups, and there are other longevity groups, so they're doing different things, but nobody's doing it quite the way we do.
One question is, so you're going to market as this large number of tests. Have you thought about going after maybe one condition or two conditions and then going all the way through the FDA and
Yeah,
becoming the standard test for that condition?
yeah, we've thought about it, but that goes back to this key point is that if we do that, then we would probably have never found the pancreatic cancer and the other things that we'll find if we follow people longitudinally if we just had the one test. And I don't think it would be successful. I don't think you get people doing a pancreatic cancer test on a regular basis if they're not getting any of the other value from the test. So I think, to Murdoch's point, people want to know not just one thing, they want to know everything. And if we can give them everything and we show them that, you don't have to worry about that, you don't have to worry about this, we got to work on. And they have the evidence so our philosophy is very different from sort of, this is why I think where Murdoch really highlighted it, it's quite disruptive. It's going to change the way we think about care. So let's not be so reductive in our approach. But having said that, we may be some of the cancer tests because they're highly regulated. If we wanted to run it just as a cancer test or maybe a broader cancer panel, I still think it's more effective to offer something more because
Sure,
everything
of course.
is related. And yeah, anyway,
Yeah,
that's
because I mean you have the answer, so why not give it?
yeah.
I,
It might be
I,
something to partner with a group on rather than doing it ourselves.
yeah, got it, got it. Um, yeah, I mean, I'll contact you, after the meeting. But let's see, any other questions? Hands raised or anything in the chat?
Yeah, guys, this
Uh,
is Yuli. This is Will. I have a question.
Will, go ahead.
Many questions, but it's hard to know where to start. But um, this is incredible, absolutely incredible. Um, so in my field, one question and one thing that I didn't see was any correlation or relation to addictions or alcoholism or things of that nature. Have you seen or do you have those types of diagnoses and biomarkers that are available?
We think so. So we haven't. So this is where we're quite excited by the behavior health and be able to break down these pathways that are probably the ones that are being perturbed. But we don't know the patterns yet. So what's driving alcoholism or addiction? Is it just an addictive phenotype, or are people self-medicating? And if they're self-medicating, why are they self-medicating? To get, they want to know how they feel because their moods are changing all the time, or they're anxious, they don't want to feel anxious, or they're depressed, they don't want to feel depressed. So there we can, our tools are very good at deciphering these. Schizophrenia is a new one that we're looking at, and when PTSD is an area that could be a combination of toxic exposures plus mood disorders and just shock to what normally or I shouldn't say normally, some people can handle and others can't, but maybe there's a combination of things going on that may all lead to helping us understand addiction. Like what's the root source of the addiction? We have it. I think the medicines like opiates are addictive. Nicotine is addictive. Alcohol is addictive. We know that, but why do we reach for them and why do we start the dependencies? Why do we go down that road? I think is an interesting question.
And why are some people prone to these
Yeah,
things, right? Not
exactly.
everybody gets addicted.
Yeah,
So why
yeah.
is that? A
Yeah,
lot
and
of
I
questions.
think it's all on I think, Will, you're touching on a point I think is very true, is that it's a it's a biophysical event, and if we could measure it, we should be able to understand what's going on and figure out how to resolve it.
Yeah. Well, is that the only question? I know you have many.
No, but I want to give everyone an opportunity. I've got a follow-up question, but just sort of
Yeah.
along those lines. If is it Are we good? Is there a line of other questions, or can I ask this one?
Go ahead and ask your second question. We
Yeah,
have
I'll go after Will. Go ahead.
Okay.
Susan is next.
Okay.
I'll make it quick. What are you guys seeing, or what are you recommending in terms of the time frame and how frequently these testing should be done? It's yeah.
Yeah.
What are you seeing there?
It's a great question. And you'll hate the answer. The answer is it depends. So in the case of somebody who is mostly healthy, an annual checkup just to make sure that nothing major has happened. We found that people are pretty stable for about 3 month period. So if you want to make changes to your lifestyle and see if you're getting effect, you want to get tested again, probably in 3 months to see if you've brought those biomarkers down into range. But then if you're getting a treatment, it depends a little bit on how quickly the drug is effective, but it could be a week or 2 weeks after to see if you've made those rapid changes or not with the medication. In the case of the behavior health we were just touching on, our sense is we want to look at somebody when they're in that toxic environment, when they clean up, so we can figure out what else is at work there that could be driving. Is it the depression, anxiety, or something else? And then what medications, and are those medications working? So in the span of a month, you could have 3 or 4 tests, depending on, what the care path is. And in the cancer, we test before treat. Is this person at risk for an adverse event? Did that adverse event happen, or is it happening? Did the drug work? It kind of depends on the length of time that it would take the drug to work. So I think there's a number of use cases and protocols for the frequency of the analysis. So it's really up to the practitioner and to the individual, and how closely they want to monitor their health.
Right.
One question, Rob, was why not combine this with genetic information, genome? I know that you're doing metabolome and proteome. Why not add genome to the mix?
Yeah,
What
and so pharmacogenomics is really powerful in this space, I think. And we did have pharmacogenomics as part of our testing. It wasn't a great user experience though, and
Okay.
meaning that you have to you get things differently, and it's a different category, but it is absolutely useful information, and I think it can be combined. And I don't think we're the best at pharmacogenomics. I think there's better companies out there. Whole
Okay.
genome sequencing, if we know the risks, you can sort of keep an eye on them. But the problem with genetics, if you're looking for disease risk, it's not that predictive for the most part, unless it's a Mendelian mutation, like a single gene that you can say, hey, this is happening now, and this is why. But the problem is it's not that actionable.
Yeah.
So, but I do take your advice, and we did try to incorporate that in there, but it was just very hard to calculate it into our whole risk analysis
Yeah.
because of the
Yeah.
timing, the unknowns.
It may not go into the analysis, but at least you can have a snapshot at the
Yeah,
beginning and see, okay, these are the 5 risks, and let's pay close attention with the metabolomics and
yeah,
proteomics. So
exactly. Well,
anyway,
we're finding doctors that are doing that in these concierge clinics. They'd like
yeah.
us to take a more holistic set of their data. I think that was a question that Susan was asking. So if they have run a genetic test and they've done maybe like a epigenetic clock, and can you combine all that and make sense of it? And we've got a few other labs here. So that's something that we have ongoing with some of our partner clinics. So those are tools that we're developing that would help, I think, with what do I do with all this information for the practitioner?
Got it. Got it. So Susan, you're next.
I just had a question about the autism. You say that you have experience with that. What is the age group? How many patients? What's your study? Just, I'm a pediatrician. I'm very curious.
Yeah, no, it was a study we did a few years back, and we've been wanting to publish it. We've been held back by our collaborator, being the university. It's just been a bit of a battle, but it's a very, very exciting study we did in collaboration with a clinical geneticist who's spent her life work is to identify the genes responsible for causing or putting people on children on the autism spectrum, because it happens so early in life. It's assumed that it's a genetic predisposition, but the demographics just don't make sense. Most genetically inherited diseases are selected against, and so for it to be a spontaneous event is extremely rare, and to have that same phenotype. So the genetics never made a lot of sense to me, and I've done a lot of genetic work myself. So what we learned, and we were able to get this remarkably highly selective, highly specific ROC curve, this ROC curve, that was really pointing to a dysbiosis in the gut. And what we were picking up were true toxic metabolites that were generated by the microbiome in the gut that were actually affecting angiogenesis, so the blood blood formation in the developing central nervous system because at birth that's still occurring. That in turn can obviously affect synaptogenesis and both have been well documented in cadavers with the people that have had autism, that the angiogenesis in various regions of the central nervous system is totally bizarre. And obviously the synaptogenesis has been documented previously. The other thing we saw were these really large and inflammatory values that in a developing brain that has a very poorly developed blood-brain barrier, those kinds of levels of inflammation can have direct effects on the central nervous system. And we know that can be very disruptive to the transmission, to the functioning, and even be compounded within the system. So that is not a good thing. And most of that was being generated by the gut. And in chatting with microbiome experts that have been studying diversity in populations, areas of lowest diversity in their microbiome have seemed to have the highest rates of autism. And so it kind of adds up somehow. Now we looked at all these different pathways.
Rob, can you repeat that sentence?
Which
The
one?
lowest what? The
Did I
lowest and the
blank out?
highest.
Sorry?
The prevalence of autism, the lowest, what population?
Well, so we're seeing we see a lot more autism in areas with where the microbiome diversity is quite low, and it's remarkable that like it's mostly a first world condition now, and often like in California, they have the highest rate of autism in the world. It's like 1 in 24 boys or something right now that is just remarkable. And so in chatting with the experts that are in our ecosystem, particularly one advocate of a diverse microbiome, Dr. Brett Finley, who wrote a book called Let Them Eat Dirt, so you can see where he's coming from, that we need to keep this diversity to have that balance. And when we lose that balance, certain microbes can take over and create this uneven system. And so, we have to balance all of those things. And so fixing the microbiome is really hard to do. We know that. Like we just don't know how to do that, honestly. But there are substrates that we found that could be restricted in the diet. If it's done early enough, we think it could actually have a profound effect. And so that's why we want to get this thing published and get it out there and work with people that are working with autism children to see if we can help avoid some of the damage that these substrates that get transformed into these toxins can have on the children.
Right, great. All right, Sunil.
Obviously, much more involved than that.
I'm sure. Sunil gets the last question. Sorry, Margaret. To answer your questions, no, it is not FDA approved, and it's a lab-derived test.
Yeah.
All right, Sunil, go ahead.
Yeah. Thank you very much, Rob and Murdoch. It was fantastic. I learned a lot being a tech guy, but and I can see a lot of science and the data engineering and data science happening into medical science, which is awesome. And I think you answered one of my questions partially, that some of these biomarkers or the symptoms per se, please pardon my language, that you are seeing, it may vary from geographical region to nationalities and to where the people are living. I'm working with a company called Septerna, where they are doing clinical trials. So we're building a platform. So are you guys also observing those kind of variations in the, if let's say you're looking at pancreatic cancer, if a patient, let's say he happens to be from Europe or from a different region, those markers or those conditions could be different, because then it will also determine that the medication has to be also different. So just curious to understand.
Yeah, it's a great question, Srini, and mostly people ask us what sort of based on your ancestral background, what sort of differences do we see? And because this is more functional, the ancestral background is not that variable across different.
Correct.
And so that's quite interesting. But you're right on. The environment has been very interesting to us. So in this, I think in this study done in Florida, one of the things I didn't mention is we noticed that there was a lot of liver and kidney dysfunction in this population associated with certain toxins, and it turns out, and I'm not from Florida, and then it is known in Florida that people do get a lot of mold in their environments, and that mold, that those toxins from the mold get into their system, and they can drive various diseases. And so, I mean, that's a very gross example. It's not as subtle as maybe, living in certain parts of different parts of the world, but it does, I think, do a good job of illustrating how important our environment is to our health and how we need to understand that in order to manage it properly. So I think you're onto something when you think about where you live and how you choose where you live. We had a patient who's not a smoker, that we had at a high risk of COPD. They had no symptoms, but they live in in an area that has very poor air quality. And 2 years later, he did start to get the symptoms, and now has been diagnosed with COPD.
Yeah. Okay. Understood. So based on that condition, then are you also kind of, the recommendation of the clinical pathway means your medication could be different because
I think well, there are a lot of factors that can be involved. So genetics is certainly another one but the nice thing about looking at function is that you take all of that into account. And so this system's perturbed. I need to focus on that system. I don't know why it's perturbed, but that's the one I have to focus on. So I think that's the beauty of the platform, is that it can take all of that information into account.
Fantastic. That's fine.
Leave your email with Ruchi, and you can continue the
We'll follow
conversation.
up.
All right.
Hey,
thank
you very much, Murdoch, and thank you very much, Rob. Thank you for patiently answering all the questions.
Yeah.
It's amazing, what you guys are doing, and good luck. And I'll follow up with you. And I'm sure there are many of the attendees that would like to follow up with you also. So we'll collect all their emails. If anyone wants to follow up with this team, please send your email to Ruchi, and she'll compile them and combine them and send it to you.
Yeah.
Thank you very much, and have a good
hosting us.
evening.
Thank you very much, everyone. It was
Good night.
fantastic. Thank you.
Nice
Thank you,
meeting
guys.
everyone.
Thank you.
All right. Bye-bye.
Bye, Dr. Yuli.